there is one specific application of DNA sequencing that works with a special protocol, a different protocol, and that's ChIP-seq. So ChIP-seq, there you're going to sequence the regions of the DNA that are bound by a certain protein, typically a transcription factor. So how you're going to do that? Take your genomic DNA with the proteins still bound to it. You're going to cut the DNA into pieces, and then you're going to use antibodies. So antibodies that target the protein that you're interested in. So what will happen is the antibodies will bind to the protein, and the DNA that is still stuck on the protein will go with it. So in this way, you can get an enrichment, a selection of the regions that were bound to the protein. Now, this is not an error-proof process. You will also get a lot of background. So these antibodies do not only bind to the protein that you're interested in, but it will bind to a whole lot of other stuff. But you will get an enrichment of the regions that are bound to the transcription factor. And from this moment here, you're going to release them. So you're going to remove antibodies and transcription factors. And from this point on, you just have to ligate your adapters. And again, you're here with a library of DNA fragments that is ready to sequence. Now, because this ChIP-seq has a lot of background, a lot of non-specific bindings, so you will also get a lot of fragments of DNA that have nothing to do with your transcription factor. Because of this in ChIP-seq, it's very important to also incorporate control samples. So this is what is called the ChIP sample. It was obtained by doing an immunoprecipitation with an antibody, but you will also have control samples. So what are used as control samples in ChIP-seq? Typically, the genome cut into pieces without any immunoprecipitation. That's one control. And sometimes people also do a selection with general antibodies. So not specific antibodies that target the transcription factor that you're interested in, but a whole range of antibodies, so pre-immune antibodies. So, and that will give you an idea about the background that is selected during your immunoprecipitation. So you always have this control sample, and often they also include a second control sample with pre-immune antibodies.